We have seen that function of integrins, that is binding of integrin to its ligand, greatly depends upon the tail region of integrin molecules or the cytoplasmic domain of the integrin molecules. There are specific molecules that interact with those domains of integrin molecules and result in its activation. But those molecules we refer to as anchoring proteins. They have to be activated by something else. So first of all, a cue or a signal has to come to cell inside in order for these anchoring molecules or anchoring proteins to become activated. This is referred to as outside-in signaling. Once these molecules are activated, they result in activation of integrins. This process is basically referred to as inside-out signaling. Let's look at this phenomena in a little bit more detail. Signals generated inside the cell can either enhance or inhibit the ability of integrins to bind their ligands outside the cell. We saw that previously also. This regulation is not completely understood. We know a few things that I shared with you, but it may involve, for example, also it could involve phosphorylation of cytoplasmic tails of integrins and association of these tails with activating cytoplasmic protein or both. Previous module we saw a case where some of these molecules interact with integrin, greatly enhancing its ability to bind its substrate or the ligand. The ability of cell to control integrin ligand interactions from within is termed as inside out signaling, because signal is generated inside the cell and effect of that signal basically results in a change outside the cell. Traditional signaling molecules, the receptor binds a ligand and changes the conformation of the receptor molecule, which is then translated inside the cell via a conformational change of the cytoplasmic domain of that receptor. Particularly importance of this phenomena we see in platelets and white blood cells, where integrins usually have to be activated before they can mediate adhesion. Remember that our white blood cells have to circulate throughout our body. They are looking for troubling issues, for example, infections or any damage to the tissue. So they have to, first of all, identify an infection, for example. In order to do that, they have to continuously move around, scanning our bodies so they can, once they find a particular problem, they have to stick there. So until they have found a problem where they need, where they are needed, they have to move freely in the body. So it will be imperative that the adhesion molecules of these cells specifically are regulated. In most other cells, integrins are usually maintained in adhesion-competent state, and that generally results in attachment of cells to the ECM. Here is, for example, the diagram shows how this procedure basically works. Here, for example, is a signaling receptor, the traditional receptor that we have talked about. And here is our integrin in its inactive form. Once the cell receives a signal, when it binds this receptor, changes occur in the cytoplasm that result in activation of this integrin. This integrin in its inactive form will not be able to bind the extracellular matrix protein, for example. Once the signal has been received, this signal is translated, it will result in activation of these integrins via modifying the structure of the beta tail, for example. That will result in activation of this integrin. In its active form, now integrin can bind ECM or another molecule present on the surface of the cell, as we have seen previously. Regulated adhesions allow white cells to circulate unimpeded until they are activated by an appropriate stimulus. We have seen that how selectin plays a role in that. Integrins do not need to be synthesized de novo. They are generally present on the surface of the cell. Once the adhesion signal has been received, they quickly become activated. Platelets, for example, are activated either by contact with a damaged blood vessel. So when platelets come across a damaged area where blood is coming out of the body, when they come in contact with that area, they basically, by various soluble signaling molecules, they become activated. And once these platelets are activated, they stimulate the intracellular signaling pathways that rapidly activate beta-3 integrin, which is found in platelets, on the membranes of platelets. This interaction induces a conformational change in the extracellular domain of integrin. We saw that schematic. Remember how the conformation of integrin changed, and now this conformational change enables the protein to bind blood clotting protein, for example, fibrinogen with high affinity. Fibrinogen then links platelets together to form a platelet plug, which basically plugs the damaged area from which blood was being lost, and the bleeding stops. Also remember that T lymphocytes, initially they are binding weakly to the antigen-presenting cells, for example, and they're basically looking at what type of antigen is present on the surface of antigen-presenting cells. They cannot bind the antigen-presenting cells more tightly. If they were to do that, they will not be able to scan the other cells. The weak binding of T lymphocyte to its specific antigen on the surface of an APC triggers intracellular signaling pathways in the T cell that activate beta-2 integrins, which are present on the surface of these lymphocytes. The activated integrins then enable the T cells to adhere strongly to the APC. So actin has been activated. Remember, actin integrins basically activation results in strong binding, and this allows the cell to remain in contact with APC long enough so that it can be stimulated. The integrins, after that, integrins have to be deactivated, so they become in inactive form, allowing T cell to disengage and, for example, go to lymph node where it will mature. During development, this regulation, integrin regulation, plays a very important role also. Cells other than blood cells also, their adhesion activities have to be regulated. If a constitutively active integrin was made, for example, by deleting the cytoplasmic tail of alpha subunit, if it is expressed in Drosophila, the embryos, for example, they have a disrupted normal muscle development. The muscle precursor cells expressing the activated integrin cannot disengage from ECM matrix, and therefore they cannot migrate normally and form the tissue that they're supposed to form. So integrin regulation is very important, not only in development, but also plays a very important role in our immune system.